Okay, so the purpose of this lab was to see if we were homozygous or heterozygous for the ALU sequence. The technique that we used was the PCR process. The PCR process works by heating a sample of our DNA until it denatures, or where the hydrogen bonds in the DNA break so the two strands are separate. So it takes enzymes we added called polymerase or Taq polymerase and builds the two new strands of DNA to replicate them. The whole procedures included swishing salt water around in our mouth so that we could get our cheek cells, and then we spit it into a cup. We centrifuged this to get a large group of the cells separated from the salt water. Then we added the enzymes like the polymerase and primers and put this into a thermocycler, which put it through the PCR process. We did this to multiply our DNA to get a larger sample. Then we added dye to this so that we were able to see the DNA after we put it through the gel electrophoresis. My DNA showed that I'm homozygous for the ALU sequence. 3 other people in my group worked, and they both were homozygous. So this shows that the PCR process actually did work for most of my whole group. For some of us it didn't work, but that could be from possible errors like the fact that this lab took over a few days instead of just doing it one day, so that allowed for kind of more contamination and stuff. But overall it did kind of work for us. Like I said, some possible errors were from contamination from how we were moving around our samples, and that the lab went over a few days. We could have fixed this by doing the lab just in one whole day. My conclusion was that I'm homozygous for the ALU sequence. Some future implications for this lab are that we could use the PCR process and the gel electrophoresis to analyze other sequences in DNA.